You're here, then we can start.
Welcome, everyone, to our second day. We're going to start off with a presentation on salmon and sailing, two great things. And then we'll look into some of our thematic reviews, hopefully move some of them along well, and then prepare for our next meeting, our teleconference, April 13th. But before we get into the meat of the matter today, I want to acknowledge Sam Harrader, who's in the audience. This is his last month in state service, and he's had really glorious contributions to the state. Quite often, state employees, federal employees as well, get horribly abused in this world, bureaucratic world, and I want to point out Sam's persistent, useful activities for many years from many offices. He's not alone in being a great state employee, but I think we should call them out, at least before they retire. Let's give them at least one good day.
But if you're sitting here, this isn't that day.
If you're going to be embarrassed, I want you to be embarrassed in a positive way at least once. So without further ado, we have Rachel Johnson and Joe Heublein from NOAA.
Thank you all for the opportunity. Can you guys hear me all right? Okay, can you guys hear me okay? Just thank you for the opportunity for inviting us here today to talk to you about some of the work we've been doing. I am Rachel Johnson, and I work for NOAA Fisheries. I also have a research lab at UC Davis. My research lab really focuses primarily on using isotopic tools to try to understand salmon habitat use and movement patterns across the landscape. I'm an applied ecologist, so I think a lot about how, in the Central Valley in particular, how water project operations and how management actions influence habitat use for salmon and movement patterns, and ultimately their reproductive success. And today I'm going to be talking to you with Joe about work that we've been doing as part of an IEP synthesis effort, where the IEP directors from the 7 agencies tasked us to develop a framework for salmon and sturgeon monitoring in the Central Valley. And so that's the work that we're going to be sharing with you today. I had an opportunity to look at the prospectus that's in front of you guys for the ambitious monitoring enterprise evaluation that you guys are taking on. And it's my hope that Joe and I, at least from a single species perspective, can share with you the process and the results of a very similar endeavor that we did. I'll be talking about winter red Chinook salmon, and then Joe will take the helm and talk to you about both sturgeon species. And so just from a process perspective, the approach that we both took was that we first developed conceptual models and looked at some of the kind of the demographic metrics that we thought were really important for salmon and sturgeon at the different life stages in the different geographic regions in the Central Valley. We then did a kind of monitoring inventory associated with kind of what monitoring was being done for the species in the system. And then we looked at the metrics that we thought were important for both science, monitoring science and status and trend in the species, as well as the management needs for the data. And we compared the two and asked the question, is the monitoring that we have in place the monitoring that we need to have in place? And we also looked at kind of some of the emerging science and technology tools that are commonly used in other systems, sometimes for decades with salmon, and also some just really innovative tools, and asked the question, the extent to which, if we applied them in our system, could we be generating more quantitative data that would be useful for both managing water resources and salmon and sturgeon in the system. So we got a lot of resources brought to bear. I mentioned that this was an IEP effort. So the directors from the 7 agencies donated to us their A-team scientists to work with us for about a year and a half to develop the products that are currently in review at San Francisco Estuaries and Watershed Sciences. So these are 2 publications that kind of we hope will be available soon to be able to document the process and the progress that we made. So we have a lot of people who participate in this effort that I'd like to acknowledge. Just to give you a sense of the structure that we used, so this is the Central Valley, and as it relates to kind of salmon critical habitat. So as many of you know, the endangered winter run salmon They spawn in one population downstream of Keswick Dam. We divided the river geographically by the upper river up to Red Bluff Diversion Dam, the middle river here in this light blue, the tidal estuary, and the ocean. And what you can see here are the different submodels we developed. So these are the different life stage transitions in the geographic regions for which they occur. And for each one of these brown boxes, these kind of submodels, we developed individual conceptual models for those. And so I'm just going to give you a quick example of what that looks like. So we took the same, if you guys are familiar with the Delta Smelt conceptual models, we borrowed from that same framework. We thought that that was a very elegant way of thinking about things that influence species in the system. And so up here you can see kind of the geographic area. This is the life stage transition. So in this example, egg to fry. And we were interested in understanding the different factors or habitat attributes that influence the abundance, survival, timing, or condition in this transition phase. So these are different hypotheses associated with what factors might influence the egg to fry survival. You can look at, red quality and quantity, for example. One of the things that we were particularly interested during the drought, which is when we were working on this, is we wanted to characterize factors such as water temperature that have direct impacts on the life stage, so direct acute mortality. But you can see that water temperature touches on a whole variety of factors that might influence the egg to fry survival. And this was a really useful format to be developing. A lot of the technical teams looking at kind of the impacts of drought on winter run salmon were able to have kind of a structured format to be thinking about the kind of monitoring that was in place in the system presently and where we might need data or further monitoring to be able to evaluate factors like the impacts of drought at this life stage. Then we were able to look at these environmental drivers that influence these habitat attributes, and as well as these landscape features. And we also took the opportunity to identify where we had management knobs in the system to influence kind of these habitat attributes. For example, substrate size, so gravel augmentation could have a positive influence on the transition between egg to fry emergence. And so we developed these submodels for each of the life stages and geographic regions of interest. And just to give you a sense, after doing this, we realized fundamentally that in a lot of the places we weren't actually measuring fish responses at these different life stages or locations. And so I'll talk to you a little bit about our summary on that. Just to dive into this a little bit more, associated with how we actually evaluated the monitoring for the different life stages. What I'm showing you here is, again, this egg to fry survival, just for heuristics to give you an example. The monitoring data we have in the system to look at this life stage transition is a really great carcass survey that CDF&W does for, it's a Cormac Jolly Sieber mark recapture model where they actually can estimate the number of females that are spawning of winter run in the upper river. And they then actually take fecundity estimates for females, and then they then monitor the number of juveniles that move downstreams 50 kilometers, so an estimate of passage. And those gear types are trawl efficiency corrected, which is rare in our system and should be commended so that we can actually get the actual raw counts and convert them to a population level estimate. And emergent from that, we get this egg to fry survival rate. And so you can see that during the drought years, we had 4 to 5% survival in the upper river. And I highlight this example because it is one of the most robust data pieces we have for winter run in the system. And in fact, if you ask anybody during the drought, any managers, how are winter run doing in the drought? They often refer to this one metric, this egg to fry survival. And here's Maria Ray being quoted as saying, only 5% survival happened in that drought year. And lower down, I have also highlighted this observation that says the number of Sacramento winter run Chinook salmon juveniles swimming to the sea has dramatically declined in recent years. So we have kind of the media saying, the number of salmon swimming to the sea has declined during the drought. But for those of you who've been up to Redding, you know that the distance between Redding, which is where this measure of egg to fry survival is pretty far from the ocean. And so there's a lot of habitat to cover over the freshwater landscape, a lot of different stressors and a lot of different things that happen to salmon in that time period. And so we went on this kind of exercise looking at where we're currently measuring Chinook salmon in the system, particularly juveniles, to assess the extent to which we were collecting data that we thought were informative to have. And so what I'm showing you here is the kind of the survey locations that we have for juvenile winter run in the system, or just for winter run in general. So here's the carcass survey up here. This is the Red Bluff Diversion Dam rotary screw trap. Here are all the rotary screw traps in the system. You've got a sack trawl, a Chips Island trawl. In the dots here are the beach seining efforts as part of the Delta Juvenile Fish Monitoring Program, and we've got the salvage facilities. And so what we did with our monitoring inventory is we invited people who were running these programs to come and talk to our group and tell us more about the programs. And we then evaluated the extent to which that monitoring program was providing information on presence. So we caught a fish on this day. So that's kind of presence and timing. Whether or not there's actually some gear efficiency being done with that so that we can actually get an abundance estimate at that location. Whether or not we actually knew that they were monitoring winter run or some spring run or fall run. So kind of run identification and the condition of fish in the system. And let me show you what we found. So graphically, for each of the monitoring locations, we then, here's just a figure that shows what that location monitors. And if you see a dashed line, it's telling you it's not monitoring that metric at that location. What you may notice is that in the upper river, we, again, where this the egg to fry survival ratio is calculated, we have the most quantitative estimate. Most of these are kind of accounted for. As you move downstream, you can see that you've got increasing kind of absence of data, and we functionally lose sight of quantitatively of how winter run are doing from when they leave the upper river to when they return as adults 2.5 years later. And so this was a really useful framework for us to kind of document the data that we thought we needed for status and trend at the different life stages, as well as being able to identify that certain areas where stressors are different in the Middle River versus the Delta, how we're not really able to capture that or decouple that or disentangle that even from things that are occurring in the ocean. Yes, Tracy?
So just quickly, it looks like condition is not measured in any of these, and this, what you're showing right here, is that true? Okay.
And and then I'll walk through each of these. So we made 6 system-wide recommendations, and I will quickly just go through them, and I'll highlight on that condition piece to tell you what's lacking and what we think would be really useful to be measuring. So we made these 6 system-wide recommendations, and I'm going to quickly just walk through those in the interest of time. So genetic run identification. So what I'm showing you here on the left are these graphs that are, this is the genetic identity of fish, broken up by these panels. So this is the real genetic identity, and these are growth curves. This is currently in the monitoring program, how the different runs of salmon, winter run, spring run being ESA listed fish, and fall run are these kind of growth curves. So in the monitoring program, the length at date is how the run is assigned. And what I'm showing you here is that these are true genetic winter run, and the blue swath is the length at date for winter run. And fortunately, many of them fall within their length at date category. Regrettably, a lot of friends disguise themselves as winter run, and the reason this is important is because winter, like there are ESA listed fish that we're trying to conserve, and then our fall run friends are ones we're trying to eat in the ocean 2.5 years later, right? So they're managed for very different purposes, and right now they're pretty blurred in our monitoring. And so, for example, there's a pilot effort this past year in 2016 where at Chips Island, so the exit of the Delta, fish that were the length at date winter run were genetically identified, and only 3 out of 44 of them were really winter run. And this has really big consequences for a lot of things, particularly if we think that it's this index or metric for production, we are wildly overestimating how many winter run fish we thought were leaving the Delta in a given year. It also influences our ability scientifically to understand the role that water project operations might be having on winter run, because we're tracking kind of the wrong fish at the wrong time. So one of our recommendations is we've got genetic tools that are being applied in many different ways, that have been used in the salvage facility for decades. We think that it should be applied to the monitoring program. The second recommendation is moving away from one fish, two fish, red fish, blue fish, and kind of fish catch, more towards kind of quantitative estimates of abundance. And so there's a lot of different ways one can do that. Fundamentally, that usually is achieved through gear efficiency tests. Russ Perry talked at the IEP conference last week about an innovative approach and method that we recommend exploring for feasibility, which is a really novel way of using coded wire tags and acoustic tags as a way of generating a trawl efficiency estimate, and we think it shows a lot of promise. One of the benefits of getting quantitative estimates at Chips Island down here and at Sacramento is, as many of you are acutely aware, there's a lot of disagreement associated with what factors and stressors are most important in terms of influencing mortality for winter runs. So there's a lot of people who say, why are you taking so many management actions in the Delta when all of the winter run die before they even get there? And so we really don't have the resolution in our monitoring program to really break up the abundance estimates or the survival between different regions to really tease that apart. And so a monitoring metric even closer to outmigration would be really helpful, just even to deconvolve freshwater impacts from marine impacts, which we presently aren't able to do. And if you think about a year like the last few years where we actually had the freshwater drought and the warm ocean blob, if very few adults return, we can't really decouple where that mortality occurred. So our recommendation is to put more emphasis into being able to generate these quantitative estimates. Our third recommendation comes from kind of recognizing that some of the best data we have for salmon in the system have come from these acoustic telemetry studies. We've learned a lot about the amount of survival that we have for our salmonid species is lower in our system than other systems. We know that each of the different runs of salmon have different behavioral patterns and migration rates. We have learned, what else have we learned? We've learned that the different routes through the Delta can have different impacts for ultimate survival of fish as they migrate through. What I'm showing you here is kind of this idea of moving away from using the acoustic telemetry as a kind of research tool, which is currently how it's used, to much more of a monitoring metric. And what I'm showing you here on the left is, I think, just really a significant advancement in the use of acoustic telemetry data. And it comes from Russ Perry and colleagues. And what I'm showing you here is survival of release groups over time of juvenile salmon. And what you can see here is a pattern in survival. And when it's overlaid with water quality parameters, that shape is kind of an interesting shape. And when you actually mirror it with water quality parameters at Freeport, it really explains the pattern we see. What you see here is when flows are low, temperature is high, and turbidity is low, you have a lot of mortality. And so being able to link these kind of from physics to fish, trying to link what's going on at the fundamental base of hydrology and hydrodynamics and water quality parameters to survival, we think is a really powerful move forward. Just a few other additional points is using acoustic tags that are real-time receivers allows for kind of real-time information to be gained from managers. So during the drought, NIMS was being asked questions associated with opening the Delta Cross Channel for water quality during winter run outmigration timing. And so the ability to know when winter run are moving past the Delta Cross Channel could afford some potential operational flexibility for water supply by having kind of tools in real time to be able to know where fish are distributed. So again, the recommendation here is one of trying to move this acoustic telemetry tool to be tagging more representative populations of salmon and to memorialize it as kind of more of a monitoring mainstay. The next recommendation has to do with looking at diversity, behavioral and genetic diversity at multiple life stages. So one of the things that we know in our regulated river is that we, using flow, have a large influence on outmigration queuing for salmonids. And so what I'm showing you here, I should say that flows really influence the way that fish queue to outmigrate. And what I'm showing you here are the results of otolith microchemistry reconstructions in adult winter run that successfully returned to spawn, and reconstructing where they were as juveniles where they spent the majority of their time rearing as juveniles. So each of these areas represents the habitat that they reared in. So you can see that the critical habitat for winter run is this main stem area here, and yet only a minority of fish are actually, that survive to adulthood, are using the main stem corridor for rearing. You actually see quite a few fish that are using the American River, these Lassen tributaries, and the Delta for rearing. And the reason this is important is because currently the critical habitat identified for winter run is simply the main stem, and we're actually seeing that a lot of these rearing habitats are that these juveniles are using habitats that aren't necessarily being managed for their protection or recovery. So we think an index of kind of this behavioral diversity and the diversity in habitat use is pretty important to capture. What I'm showing you here on the right are these survival landscapes from Keswick Dam based on temperature mortality downstream for different years. And you can see here that during the drought, this temperature in 2015 was incredibly hot for winter run eggs. And you can imagine that in different years, water operations play a pretty heavy hand in determining which of the winter run might successfully reproduce in a given year. And so we think it's pretty important to be using kind of parentage-based reconstructions to be able to understand how many female winter run, like the effective population size, are actually contributing to the population every year, as well as linking how water project operations might be influencing the ultimate reproductive success. The fifth recommendation out of the 6 is condition. It's something that is not routinely monitored anywhere in the system. There are kind of these episodic studies where this information is being gathered. One example of this is Scott Foote's work during the drought, where they put these sentinel fish, they're kind of like clean lab rats of salmon that you put into water, and then you monitor their virulence of disease. And what he found is when he placed these sentinel fish in kind of winter run upriver habitat, that 86 to 94% of them were infected. And so we think that potentially in many years, but certainly in this particular drought year, that disease might be an important factor that we're not really consistently monitoring. And then also related to condition, one of the biggest likely changes that we're going to see in the Delta in particular is habitat restoration. We think that this might also be important for influencing abundance, but more primarily it'll probably influence the quality of fish that come from the freshwater landscape that we produce. This is a photo from Carson Jeffries on the managed agricultural floodplain work on the Yolo Bypass. We're looking at these two different sized fish. They're only 4 days apart in age, which really speaks to how different access to different habitats can play in the growth rates of fish. And work that Lindsay Woodson and I published shows that the growth rates on the freshwater habitat have a strong influence on their ocean survival. So in years where ocean conditions are really poor, fish that look like those bottom fish have a better competitive ability and survivorship probability than the fish that didn't have good growth opportunity in the freshwater landscape. So the recommendation here is that interannually, like a year like this year, you could imagine that the quality of fish that we're producing might be fairly different than what came out during the drought. And so we need to have this kind of annual check-in or metric about the quality of and quantity of fish that are coming from the freshwater habitat. And lastly, I think there's going to be rapid progress. There's already been rapid progress in making data available. So for those of us who do technical synthesis work, it's been incredibly difficult to work on salmon synthesis work because most data sets live on local computers from people who are generating and collecting that data. What I'm showing you here is a table of the different monitoring programs that I showed you in the monitoring inventory, and the extent to which that data is available on an open source platform that can be data hubbed and crawled, like on Bay Delta Live, or can be grabbed for synthesis work. And you can see that the majority of them are not available. There's been some rapid advancement with SACPAS, which is work that Josh Israel and colleagues at UW are trying to make this monitoring data available. So this table is no longer updated. Both Tisdale and Knights Landing are now providing their data in an open access way. And the IEP, I'm just going to give a plug there, or get an A+ for continually providing that data online. And so this has been a recommendation for the monitoring program, is to make the data available. And so just in conclusion, I hope that I've shared with you kind of the slice of a single species approach to of how we assess kind of what the monitoring needs are for status and trend, and that we think would be useful for informing both life cycle models and synthesis work relevant for managing the species. And these are kind of the 6 system-wide recommendations in summary that we recommended. And I'm going to just turn the focus over to Sturgeon, and then Joe and I have a conclusion slide and can field questions that you might have at that time. So with that, I will hand over the baton to Joe to talk to you about Sturgeon.
So I'm Joe Heublein. Let's see, I better turn this on. Okay. I'm Joe Heublein. I work for NOAA Fisheries here in beautiful downtown Sacramento. My talk's going to be a little bit redundant to Rachel's. Reading your monitoring prospectus, the monitoring assessment prospectus, I decided to describe the process of our monitoring evaluation because I think it has actually a lot of application to this effort that you're going to conduct in the next year. So, given, I think, 6 presentations on this, and I realized I needed to kind of step it back to the 7th grade book report. So, say what I'm going to say it, and then say what I said. So, I got 4 main objectives. I'll just read those, and I'll be touching back to these as I go through this presentation. So, our Sturgeon team objectives were to identify questions in sturgeon management, determine the extent and performance of the existing monitoring enterprise, develop conceptual models illustrating potential studies that addressed outstanding questions, and provide general monitoring study recommendations based on 1, 2, and 3. So these were our 4 fundamental steps. So Rachel gave you that long list of agency scientists that were involved in this process, so we were kind of in a unique position to identify these key management questions. They basically fall into these 2 general categories. You've got population status and trends and mechanistic studies. Population status and trends are basically abundance and life stage abundance, maybe trends over time. Here's a draft green sturgeon recovery plan. We have demographic recovery criteria. Here's a 2015 harvest estimate, if you will, or the number of report cards that were sold in the white sturgeon fishery. If you multiply that number by 3, that was the authorized harvest of white sturgeon in 2015. Here's the Iep Science Agenda. We have a lot of general life stage monitoring recommendations in there. So with these, a lot of people maybe this isn't very glamorous monitoring. It's just, okay, how many fish are in this particular life stage or how many fish are in this population? But it still provides the foundation of the work that we do. And it's not necessarily useful unless you actually put it in context. And the context is the mechanistic studies. So why is the abundance high, or why is the abundance low, or how are these fish going to react to a particular project? Here I've got the cow water fix. That's something that is on everyone's mind. And here's just a general figure showing an adaptive management process that we included in our SAIL manuscript. Okay, so moving forward, we moved to develop a monitoring inventory. So that's number 2 on that first slide way back there. Again, you have these 2 general monitoring categories, and we actually filtered even further into 4 subcategories. We have life stage surveys, that's the long fin. The base study, Otter trawl is how we generate a juvenile white sturgeon index annually. Tissue analyses, that's a sturgeon from a fishing derby that they're analyzing the reproductive condition. Population modeling, that's actually a graph of the white sturgeon juvenile index from the base study, and telemetry. So I'll go into these in a little more detail in the later slides. So I think it's worth noting that each of these studies has their specific application. So, you're not going to get an abundance estimate from that contaminant study you see right there from a sturgeon derby, where you're taking ovary samples, and you're not going to get in the converse, you're not going to get an abundance estimate from, or sorry, you're not going to get a contaminant estimate from the Bay study. So you need all of these things to have a complete monitoring program. So real quick, after we went through this inventory process, we separated all the existing monitoring into these 4 monitoring categories that I described, and also by sturgeon life stage, which I'll get into later. So that's step 1 and 2. Identify the questions, see what's being done, and see what's possible and feasible in future monitoring. So you have this management question, and you have a inventory of monitoring. You need to have a framework for organizing all of this information. Obviously, these are broadly distributed fish, British Columbia or Alaska, all the way to Baja, Mexico. Many monitoring programs have some interaction with these fish, and we can't just put all of these questions and all of these management and monitoring recommendations on a piece of paper and say, hey, here you go. So I've sort of realized through this process and through many revisions of our manuscript that a conceptual model is necessary to organize this information in a way that people can understand. So anyway, our conceptual models were organized by life stage, as I kind of described earlier. You have eggs, larvae, juvenile subadults, and spawning adults, and also by geographic region, similar to the Chinook conceptual model. Upper, middle rivers, estuary, and ocean. So that's 1, 2, and 3. Questions, inventory, and conceptual models. So our first recommendation was life stage surveys. This is the foundation for all of our fisheries research. And here's sort of a example of the process that we would go through to evaluate how we are doing in a particular monitoring category. Here's green sturgeon, existing monitoring in the system. So I went through and actually graded how we were doing for actually evaluating abundance and trends at these life stages. So you got egg mats. Right now we are surveying egg mats in the Feather River, and the Feather River, green sturgeon have spawned in the Feather River once. So it's not their consistent spawning reach, and that is the only area where we are doing any form of egg sampling. So I give them a D- there. I haven't. Don't tell them. Don't tell anybody I said that. Next, you have larvae. Larvae are collected in rotary screw traps and D-nets. Although we don't conduct rotary screw trapping for larval sampling in the extent of the green sturgeon spawning range, we have a good monitoring site right smack dab in the middle in the form of the Red Bluff Diversion Dam screw trap, and that's been ongoing since 1995. So you've got a long data set, and you can potentially extrapolate out from that to see how larvae are doing. So I give that a B. It's not completely, it's not comprehensive, but it's pretty good. Next, you have juveniles. There's Benthic and Otter trawling, gill nets. Until this last year, we've had a real hard time catching green sturgeon in any of the surveys, and actually haven't really been trying. So that's a C. And adults. Trammel and gill netting in the estuary for sturgeon has been ongoing in IEP since the 1950s. So this is the most consistent sampling of green sturgeon in the entire system. And although in some years catch is really low, at least we have a data set and something to relate to a time series. finally, we have kind of our flagship, our only abundance measure for green sturgeon is the Didson spawning surveys. I give that an A- just because that has been someone's PhD dissertation for the last 5 years, and we've been cobbling funding together for our only abundance measure of a threatened species. So, to give it an A-. But I was kind of going through this last night, and I realized you need about a 3.0 GPA to figure out how fish are doing. So we're not quite there, and again, this kind of forms the foundation of most of our recommendations. Population modeling needs this data, telemetry needs this data, tissue sampling needs this data, etc. So I think in that way we sort of depart from the Chinook model in that we just have these broad recommendations that are a starting point for moving forward. So anyway, you saw that alphabet soup of grades for the life stage surveys. I was thinking it kind of reminded me of my college transcript after I took organic chemistry. Anyway, thought that would be funnier for you guys. Yeah, exactly. Yeah, exactly. So we have the small encounter rate with green sturgeon, and we have to develop technologies to make this data more efficient. And we've been moving forward and had some great advancements on genetic analysis. You have long-lived fish. There's a high likelihood that you're going to interact with a fish or a fish's relative over time and be able to model a population with those techniques. And you have aging of pectoral fin rays. And this is something that people have been doing in sturgeon biology for 50 plus years. Basically involves taking a pectoral fin ray biopsy, estimating the age, and trying to back calculate periods of strong recruitment. So if you don't have a actual juvenile recruitment index, but you have a survey that contacts a lot of adults, then you have an opportunity to potentially back calculate or model when recruitment occurred and what the management conditions or environmental conditions were in effect during this period of recruitment, and maybe try to simulate that with future management. So again, on the recommendations, there's telemetry. In some ways, it's uncertain whether we are going to be able to get these abundance measures at each life stage, and we may need to base management decisions just on individuals. There's been some pilot efforts if some of you were at IEP on tagging juveniles and seeing how juveniles potentially react to some of the conditions in the delta. That picture on your right, I don't know if that's on your right or just my right, but anyway, that's a sturgeon that's being satellite tagged after it was caught in a commercial fishery bycatch. So these are ways to essentially expand the value of a small catch of fish in these surveys. And something that we had in common with the winter run effort was a need for long-term support of these acoustic arrays and maintenance of an accessible array database. Sturgeon are somewhat unique in that we need 2 arrays essentially to do this work. Large sturgeon, the large sturgeon tagging enterprise or adult sturgeon tagging enterprise along the entire West Coast uses a particular technology, and it so happens that most sturgeon are tagged outside of the state of California, and we rely on data from those fish entering California, even to estimate our run size from that Didson survey. So not only a Chinook or juvenile fish acoustic receiver array is necessary, but also an adult sturgeon array. So I think I've sort of touched upon this a few times, but there's also population modeling and synthesis. There's a need to develop and review the existing recruitment indexes and harvest and abundance estimates. I've got a recruitment index from, let's see, what is that from? I believe that is from the Bay study. No, I take that back. That's actually a salvage. Those are salvage estimates. Even how we interpret these salvage numbers. Jerry Morinaka gave a presentation on what a small data set some of these large salvage estimates are based on. So some of those 400 or 500 fish salvage estimates are based on like the collection of 2 fish. So, and this, if you read a lot of the documents on the status of green sturgeon, a lot of them rely on this salvage information, and we haven't done a thorough evaluation of what it means. So, again, another thing we have is harvest estimates with reward tags and angler reporting. These require some pretty involved quantitative techniques to understand angler compliance with reporting and tag returns and so on and so forth. And green sturgeon are also a challenge in that a large percentage of the population may exist outside of the state of California. So abundance modeling requires a multi-state synthesis. This is a table from past commercial harvest in Oregon when it was permitted. And right now, there's some estimates of the green sturgeon southern DPS population at around 10,000 adults and subadults. And this particular area has a mixed stock of northern DPS and southern DPS of about 50-50. And that y-axis is in the hundreds of thousands of pounds. So in any given year, you had large harvests of green sturgeon outside of the state. It's just an indicator, among other things, that we have to be aware of a large proportion of the population being out of state. So this was from my IEP presentation. This just shows the monitoring studies, mainly in IEP, that have opportunities for recruitment, indexing, tagging and tissue analyses, and synthesizing information for modeling. So I'm going to give you a quick recap. So we generate those management questions, and a easy conclusion was that answers to nearly all of them rely some degree of life stage surveys. Monitoring inventories are necessary to establish the context and feasibility for testing hypotheses and questions. Conceptual models provide a framework for illustrating and organizing recommendations to address these hypotheses. And I guess my final point is that monitoring alone will not produce a single sturgeon. So right now we have some pretty crude knobs on sturgeon management. I think that you could probably show a response with harvest, and you could probably show a response with a large-scale passage improvement or habitat access improvement project. But that's it. And so although monitoring won't produce a single sturgeon, if we can effectively monitor fish to actually understand how they're reacting to some of these smaller scale management actions, that may in turn have a population level effect. So with that, I think we've got the last slide with Rachel.
So I just wanted to take the opportunity to kind of think a little bit about the process that Joe and I underwent, and thinking about the process that you guys are about to embark on, and thinking a little bit about if there are things that we kind of learned from our process that might be useful for you guys to circumvent or to think about as you're moving forward. The first one is the geographic scope of what you're looking to take on. So in looking at the prospectus, it looks like delta monitoring is what you've taken on, and there's been a lot of progress, I would say, at the IEP director table, and not just thinking of the legal delta, but for salmon and for sturgeon in particular, to be thinking what they would say is Sierra to the sea. And I always like to say Sierra and including the sea, because a lot of the salmon spend the majority of their time, and we've heard from Joe that our green sturgeon friends spend a lot of time in the ocean, and it's not something that we really think a lot about, kind of on the inland side of things. And so if you're thinking about trying to include if you're thinking about trying to include kind of salmon and sturgeon in your assessments, thinking more upstream of just the delta, I think would be really useful. One of the challenges that we had in our own group in thinking about how to constrain things, this was an IEP ask, which also really focuses primarily on delta monitoring, is that a lot of the salmon monitoring is really fragmented across the landscape in terms of who's implementing it, who's funding it. And we as scientists decided we're just going to put blinders on all of that, and we're going to ask what data do we really need holistically to better manage salmon from the life cycle perspective. And so there's just kind of that balance between your assignment, the scope of what you're taking on, and how to implement it. And the similar type piece is that when we were looking to think about the monitoring enterprise, we kept thinking, well, is it really an IEP product that we should be, is it really an IEP thing? And we kept thinking, whoever implements the recommendation, that's a separate thing. We need to actually document what is needed. And so I guess I would encourage you guys to think about kind of upstream pieces to the delta, as well as downstream, depending on what your kind of metrics or ecosystem metrics that you're looking at might be. Compliance monitoring and the new regulatory horizons. I think some of you may have met with Erin Forsman from EPA yesterday, and I think she's very inspirational, and I think she sees a lot of opportunities on the regulatory landscape for thinking about monitoring needs on a broader scale, with the State Water Quality Control Plan being updated, with Cal Water Fix Adaptive Management, with Eco Restore, with the reconsultation of the projects, the life cycle model and its use, a lot of the winter run data that I showed you, would really benefit the life cycle model, which integrates how population dynamics in winter run are influenced by water project operations. And so there's really an exciting opportunity, I think, for you guys to play a fundamental role in advising on what kind of monitoring needs to be done in the delta and for kind of key managed species in the system. I would also like to share this observation that I've been told many times myself. Because monitoring is so unglamorous and unsexy, it's not like you just built a bridge and just get to show it off, or it's not this glitzy tool. It's kind of long-term fiscal investment, looking at status and trend, getting a new index maybe annually. I've been told that the only kind of science that gets done is called compliance monitoring. And so there's just the reality or advice that I've been given in terms of thinking about the role of how science is funded and done in our system, and that compliance monitoring is commonly what is funded and done. The last one is we looked at kind of emerging science and evolving technologies from other systems. You guys have amazing lenses in the geographic range that you guys work in, and on the kinds of disciplines that you guys bring to this exercise. I think it's really exciting to think about what you guys might be contributing as a collective in that way to this local system. It's a large endeavor to try to compile monitoring inventories. That's going to take a lot of the time. And I think there's a lot of value in compiling it. There's actually a lot of documents that have already done that. So looking at the IEP, Honey et al. 2004 looked at all of the IEP monitoring in terms of what species are being monitored. I think the real value from your efforts will come in the way of defining what those kind of ecosystem performance metrics are that we need to achieve with monitoring, and having a finger on a heart rate on the delta, and coming up with advancements for how to measure those quantitatively over time. The other thing that I was thinking of as Joe and I really looked at a lot of the monitoring from a single species perspective, and one of the challenges we find is that a lot of our existing monitoring are intended to be community surveys. So what we get to what we offer was a single glance. And so, for example, the beach seine sampling that a lot of effort gets put to did not come out high in the salmon world for being really relevant. At the same time, it's incredibly important for a split tail index, right? And so just not being short-sighted about what existing monitoring is providing for what you think it needs to provide, but if you removed it, what might you be influencing by doing that? And also thinking about species that aren't listed yet and the data time series that might be being provided by some of these other monitoring programs. Joe, did you want to share any other kind of insights from what we took on?
Okay. I think just a final point was that we did a two-species evaluation, and in the process of doing that, I stumbled upon some other multi-species conceptual models, and this also followed a similar format to Delta Smelt. So I think something to keep in mind is that you can just transfer species names and potential environmental drivers and hypotheses around, and this is a useful format for evaluating monitoring across the board. So in that regard, if you're going to be evaluating water quality sampling or something along those lines, you can literally use this type of format and save some time in kind of building off of all that past work. So
Great, thank you. This very thought-provoking discussion topic. I think there are probably a lot of questions.
Yeah, that was very interesting. I really enjoyed those talks. And one question about maybe it relates more to the salmon inventory of the monitoring programs. Did you focus primarily on the salmon monitoring and the ancillary water quality kind of measures that were taken alongside with the fish sampling, or did you look at comprehensively all of the various parameters you talked about in the one slide, for example, with the eggs to fry, all the monitoring, water quality types of monitoring that are being done?
It's a good question, and we actually we started on this pathway actually trying to choose a topic that we wanted to do some synthesis effort under. And as we developed the conceptual models, we realized that we weren't able to do some of the synthesis work we wanted to do because we weren't actually measuring a lot of the fish variables in a quantitative way, just the fish responses, like at the top of what we think all of these things were influencing. So it really focused us to go down this kind of monitoring inventory pathway and more on the fish response side. I think the upper river framework, in our evaluation, we did not formally look at the other monitoring data sets that are occurring that could be used with the fish pieces. So we did not evaluate all the habitat attributes and ask questions like, are we actually measuring the quantity of spawning gravel? We did not systematically go through and see where we had data available. But that's certainly something that you guys would probably want to do with the conceptual models that you develop, is kind of piece out each one of those and ask the question the extent to which that that monitoring, water quality monitoring is being collected. We did not do that, but recognize that the upper river conceptual model that we developed during the drought was an amazingly useful framework for asking where supplemental monitoring needed to be done. So people were like, we don't know anything about dissolved oxygen in reds. Do we need to know anything about it during the drought? And so it asked, it highlighted, oh, are we monitoring this? Do we need to do more monitoring there? So it provided them all there, but we did not actually review it in our effort.
Yeah, sort of a follow-up then is, because then did that evaluation give you any better insights into what kind of time and space scales those other kind of monitoring ought to be done? I can see in some respect the space scales being answered, but the time scales, I know the biological time scales are easy enough, but what about the drivers of ecological changes and stuff? Any insight into that, or did you think about those?
We thought about those. A lot of the kind of abiotic data, meaning kind of river temperature gauges, turbidity, those existing gauge metrics are available along that kind of geographic space, but we did not look to see if there was like lower trophic food web sampling or chlorophyll. So we did not kind of evaluate those things, although those things would be really important in the context, especially of trying to get a better handle on kind of the mechanistic pieces associated with why you might see changes in any of those fish metrics. That would definitely be the place to start looking at kind of the mechanistic studies to better try to understand what's driving those changes.
Okay, thanks.
I think just on the sturgeon side, we didn't actually inventory things like temperature and flow and where they were measured in geographic regions. But going through the process, and it's kind of buried in the report a little bit, there is a pretty coarse resolution in the upper middle river on some of those abiotic factors, and having to go through a modeling exercise to figure out what temperature and flow is in a particular spawning reach is a little less efficient than having improved gauging data. So I think that was one thing that we realized, at least on the sturgeon side.
Yeah, thanks so much for coming and making this presentation. Tracy asked the question about condition. So you talked about condition indexes are not calculated, or indices are not calculated. And then you talked about parasitism and habitat influence. I mean, there's so many old, 50-year-old condition indices based on, length and weight. I mean, they've never been done?
No, it's one of these amazing things where I'm like, Fulton's condition factor, do we even have K? I mean, we don't actually, the fish aren't weighed in the field, in the monitoring programs. They're just not.
Yeah, I've got a couple of perhaps broader questions. It seemed when you were going through the monitoring by life stages that of necessity there are different methods used for each life stage. And the question then is, how do you cross-reference those methodologies? How do you know that a particular value for one life stage bears any relationship to some value for another life stage gained by a different kind of sampling methodology? Okay, I guess that answers it.
No. What I was going to say is the primary gear that is used to. So, in my mind, I'm thinking of the difference between smolts and fry and how those are kind of different. I don't know if you were thinking kind of more broadly associated with kind of adult, egg, and juveniles. Were you asking more refined in terms of the juvenile life stage monitoring?
Well, I was just noticing in general that you listed the different life stages and the methodologies that were used for monitoring those, and they're different, understandably. But you would like to know what the cross-referencing is, whether if you're going from A to D, for example, are you in fact, what are the translation factors that come into play?
I think therein lies the conceptual model and maybe not necessarily conceptual model, but the population modeling and how those abundancies relate to each other is kind of a broader quantitative modeling exercise. But at this point, I think that there's some intuitive or anecdotal relationships. But definitely, if with a complete life stage monitoring program, you would also need to know what 10,000 larvae, how that's related to 5 adults. And there are certainly methods to do that. And given that, at least on the sturgeon side, we don't have a really extensive monitoring network, no one's necessarily taken that process on. But there are some things like that out there.
Well, it impresses me that as we begin to look at monitoring over a broad area of reference, that this is an aspect that we're going to have to consider. There's another aspect of the same sort of thing, which is the assumption that the efficiency of sampling, or the flip side, the biases in sampling, are going to be constant among years and places. So if you look at a drought year versus a wet year and you see different numbers, how much of that is due to the different numbers and how much of it is due to inherent biases that come along with the sampling methodologies and their effectiveness when you got a lot of fish versus a few fish?
So from the salmon side, that gear efficiency piece associated with covariates is incredibly important and is actually fundamentally one of our recommendations. So right now we don't really have a quantitative ability to look at between year comparisons because we don't actually have an abundant, like a demographic abundance estimate that's calibrated to the gear that's collecting it, right? We just have fish catch. And so there's a lot of effort associated with, like at Red Bluff Diversion Dam, that rotary screw trap has classic gear efficiency with flow relationships, so that as a function of flow, that gear is calibrated to then give you the abundance estimate. We're trying to do similar work with doing calibrations with the trawls associated with other covariates, tides and turbidity, and so really trying to understand the catchability of the different gear types so that we can actually come up with a metric that's robust to be able to compare between years and really variable hydrologic regimes that we see in the system. So the goal is to kind of come up with these robust quantitative estimates using information associated with the variation in trawl efficiencies and kind of nailing that piece more refined.
Yeah, it impresses me that there's almost sort of a catch-22 involved in this, that if you've got this sort of intensive data collection and a data management system that you can use to probe it, that you can begin to get at these efficiency, effectiveness values. But getting that quantity and quality of data is itself an expensive proposition. So if you're going to talk about monitoring broadly over the delta as a whole, it's not likely that you're going to be able to gather the information on everything to develop the kinds of correlations to examine those sorts of relationships. There's no simple solution to it. I think it's just a reality that we have to recognize that there's a problem there.
Yeah, I think for fish sampling, it's a little bit more straightforward. I say that. Not knowing the full breadth of different metrics that you're thinking about when you say that. But in other systems, gear efficiency estimates are routine. Like one wouldn't put a rotary screw trap in a river unless you actually were able to get a population estimate with that gear type. So in other systems, and salmon are kind of an easier species than others to be able to do trap efficiency estimates with. I mean, delta smelt, good luck. And if we can't nail it with salmon, then we should be able to nail it with salmon.
I had one other sort of philosophical point, which is not really answerable.
That's going to be you're going to take that one.
Before you get to that, I was going to say really quickly that over time we might realize that these life stage monitoring surveys co-vary. So you can drop eggs and just do larvae, or drop juveniles and just do subadults. So I think that would probably be the ultimate goal to improve efficiency of the overall program. But that would obviously take time with an actual effective monitoring program to develop those relationships.
Yeah, you have to have the data in order to determine how do you trim the data to monitor just what's going to be most effective and cost effective. The other point really is something that affects any kind of measurement of environmental systems, and that is the fact that you intervene in the system when you measure it. And therefore, what you are measuring is a combination of what is there and the product of your intervention. It's the Heisenberg effect on ecological sampling. Do people in your arena think at all about that? What effect? Obviously, if you're going and doing a trawl, things like that, there's likely to be some effect that would not be there had the trawl not occurred. But is there a way of assessing that sort of factor?
If a tree falls in a forest, nobody's there to hear it. Yeah.
Okay.
I don't think anybody's.
As I said, a philosophical
Yeah,
point.
no, it's-
But
it's
I
a good
think a
point.
reality that we will have to think about.
Agreed.
A lot. Yesterday, you mentioned when we were talking with Aaron Forsman about the monitoring prospectus in a smaller meeting. One of the things we talked about a lot was this problem that compliance monitoring is what you're going to be funded to do, because that's what you're going to be required to do. And how do we move from compliance monitoring of fairly small, easily measured pieces to something that's more environmentally relevant, more of an environmental outcome, if you will. And one of the things that occurred to me, and I'm just curious what you guys' thoughts are on this, the issue of condition index is something that isn't being measured. There's so much in the Columbia showing that condition index or lipid content is, I wouldn't say strongly predictive, but at least predictive of first overwintering survival for outmigrant juvenile Chinook. So I'm just curious philosophically, to what extent are you folks thinking as you think about trying to have a more, if you can get enough data, get more efficiency in monitoring, do you foresee the ability to eventually, in your careers anyway, have compliance monitoring that is actually more environmental outcome based?
I think that you guys could play a heavy hand in the opportunity for that. I think that that is a direction that that would be very advantageous to move forward under kind of the compliance monitoring umbrella, to be able to assess how fish and the system is responding to climate change, kind of these larger scale influences, to be able to then even look at kind of project level effects, which is what the compliance monitoring is really intended to look at. I think that there's going to be a need for more effective, an assessment of effectiveness monitoring associated with actions. And I think that moving forward in kind of the adaptive management pathway that it seems like a lot of the regulatory agencies are moving forward with, I think that gives the opportunity to actually redefine what compliance monitoring really has been defined to include.
Okay. And then one small question I have to ask, on the slide showing the water quality parameters versus survival over different days, it was a progression of dates. What survival was, from 0.6 to 0.2, so 60 to 20%, but it went up and down. So explain to me what that metric, what was the y-axis on that slide? Because how do you go down to a 20% survival and then survival goes back up if you're measuring the same cohort of fish? I had trouble with organic chemistry too. I had a lot of trouble with statistics as well. I had trouble with almost everything. So I'm dumb, so just explain it to me.
I'd have to go back through it, and I think it's reach specific survival, and it's the route.
Okay, so it's reach specific survival. Okay, that covers it.
And then
yeah.
through delta survival is the overall purple, so it explains kind of the overall.
So if you had like a 20% survival for 2 or 3 reaches, that means you're now down to like
A few percent.
1 out of 1,000 fish or something. That's what I'm trying to figure out is if it's a cumulative sort of, if you have from 60 to 20% survival and you got like 20 or 30 points, you've got almost 0 fish, it seems. I don't know.
Yeah, I don't think it's cumulative, but I'd have to take
Okay.
it back. I'd have to actually
Okay,
look at
okay.
Russ's work and actually reconcile that question.
Just remember me and chemistry and everything else. So
No, I think I should be able to speak more fluidly to it, but I haven't thought of it with those lenses.
Actually, I had the same question as Tracy, and because that number is kind of very confusing, because it was only a 3-day difference in timing and getting in those survival rates. Just didn't make sense to me that there'd be that rapid of a change up and down. Anyway, more broadly though, talking back to, again, Tracy's just question about compliance monitoring, would you think that is the agency fish sampling that's done out there, is that broadly categorized as compliance monitoring? Somebody's requiring you to do it. Do you consider it compliance monitoring? Does IEP consider it compliance monitoring?
So there are, so it varies. What I can say a lot of monitoring that's done that we find useful for salmon are kind of the long-term surveys that were initiated through D1641, so they're actually water quality permit monitoring. So they're not necessarily part of the BIOP for salmon monitoring, but in the biological opinion, there is language in there that does endorse the Delta Juvenile Fish Monitoring Program and the data generated for salmon under that program as compliance monitoring, as thou shalt continue doing this so that we can monitor salmon. It doesn't have a lot of specificity in its language. It doesn't say, and you need to trawl at Chips Island this many days, and we need this metric from it, but it is present in the kind of biological opinion. So thus, I guess it would be considered compliance monitoring.
Because we heard a number the other day of $22 million spent in monitoring, and 20 million of that was compliance monitoring. And I'm just wondering if is the fish sampling, for example, is in that broad category of compliance monitoring, or maybe it's not classified that way. But
There's differing opinions too on what is compliance monitoring, just because some of the language is so broad. So you'll have very specific things identified potentially in a biological opinion, and then there'll be some general discussion. But I would say that's a good question of if all of the fisheries monitoring is included in compliance monitoring. I would think that it is in that figure, but having more specificity on what exactly is long-term compliance monitoring and sort of agreement amongst agencies would be a potential improvement.
Steve, I'm very glad you asked that question, because this is one of the issues that we're trying to deal with, the flexibility in compliance monitoring to make this continuum with what we call environmental monitoring. Because Steve, perhaps you could comment when we have the public discussion on this too, because this is really a very big issue in terms of actually moving to environmental-based outcomes. And the figure Steve used of 20 to 22 million, I mean, we're hearing that all the time. But again, if it's flexible, then that really is quite different, which is what we're trying to get at with this review. So thank you for bringing that example up.
And I think it's in a chicken. I think we're in a time where it's a chicken and an egg, too. So in some ways, something might be called compliance monitoring, stated in a water quality document that's now being revised, or biological opinion that's now being revised. And so the extent to which there is something that you guys could provide as recommendations for what you think you need to have for the heartbeat of the estuary, that can then be looked towards as a recommendation for what a holistic monitoring program under adaptive management might look like. Does that make sense? I wouldn't restrict yourselves to just looking at what's been classically defined as compliance monitoring, but rather I think there's a lot of value to including the environmental monitoring as an emergent, if you think that data is needed, then that gives a lot of power to those who are trying to memorialize data that's needed to be able to get the heartbeat of the Delta kind of in the right processes moving forward.
I noticed you mentioned genetic DNA monitoring, essentially, and it kind of surprises me, just like it's surprising that they don't weigh the fish at different locations when they're sampling them, that there's not more genetic data collected and made available. Certainly our common friend Mike Miller would like to get access to a lot of that stuff and would do all kinds of statistical magic with it in terms of populations and carrying over of populations from year to year. There's a lot you could learn, I think, perhaps from that, at least in theory. What are your thoughts about these different new kinds of monitoring and their potential for improving our ability to do this kind of work over time?
I think there's a tremendous amount of potential, and I think that even on the fundamental level of knowing we're at a very fundamental level and trying to incorporate just modest technology into the monitoring program, meaning knowing whether or not it's a winter run or a fall run fish in our monitoring is pretty fundamental. I think if if people couldn't tell the difference between a delta smelt and a longfin smelt and monitoring, people would just be, oh my gosh, we have tools to tell them apart. Why aren't we actually doing this? And we don't find that same kind of up in arms nature about not knowing which salmon we're interfacing in our monitoring. So I think that there's some fundamental needs for these new techniques. I think we should always be looking for science innovation to do a better job of providing quantitative data. So our objective in the sale process and manuscript was to come up with these high-level recommendations. So our recommendation, for example, is to get an abundance estimate or to tie it into more of this demographically relevant rate like survival, recognizing that as technologies improve over time, there might be a better way of measuring that, and we shouldn't stifle monitoring in this archaic way, like we've done it this way, because we're always going to do it this way, kind of more of an index-based approach. But if you link it to these quantitative population-relevant metrics, then you can evolve with new technologies seamlessly and not have to worry about whether or not you're cross-compatible in the back. So I think, yeah, that intent is as new techniques emerge, and if you've got the right metric that you're trying to follow, then it's really robust to that science innovation.
Yeah, I was very impressed with your length by date scattergrams, that would indicate that you probably have a very high probability of misclassifying winter run salmon, and therefore what we think we know about winter run salmon might very well not be what's really true for winter run salmon.
And one of the observations that we've had is that we're generating these data sets, but we're very limited or cautious about using them. So for example, in the life cycle model, not knowing, so the life cycle model uses data at Chips Island only for 4 years because those are the only 4 years where there's genetic identities used in it. So one of the challenges we face is that we're collecting all of this monitoring data, but we're actually not feeding it into a lot of these synthesis processes or these more quantitative processes because they're not providing the quantitative data that we need.
Well, go ahead, Joe.
Being a non-fish person, I really enjoyed the presentation. Thank you very much. But when I look at this, the life stage surveys, you are generating data. Now, you are unhappy with certain things like the egg mats, you get a D minus. Is there any kind of process being now implemented so you can get kind of uniformly informative data on this?
I think one of the benefits of this process is how many agencies were involved, and so people have been reviewing drafts as they're finalized, and actually this year there's some plans to do more egg mat surveys, for example, and I think on the salmon side and the sturgeon side, people have reached to some of the recommendations and started implementing them before anything's kind of been finalized. So that's been a benefit. But some of these surveys are pretty expensive and require a lot of resources, and it's questionable whether people will be able to sort of reallocate what's existing in maybe IEP, for example, to improve some of those grades.
And I would just like to add that on the salmon side, we've actually had a tremendous amount of success in actually moving the needle on implementing some of these recommendations. I think it had to do largely with it being a multi-agency team that was together, so people then were able to go back to their respective agencies that were being kind of evaluated and make some improvements. There's been some, I think, there's been some increased financial, there's been some increased funding to try to see some of these recommendations implemented. And so I think there's a lot of value to what you guys would put together in kind of creating that vision and putting it together. Then people, as fiscal resources allow, they can then say, oh, well, this is something that has a lot of multi-agency support. The fish people think this is a fundamental data gap. Let's try to pull in the same direction with these available funds. So I think there's a lot of value in what we've done in terms of providing some guidance on where we think some of the bigger data gaps lie.
Well, thank you very much. This is a great, I would love to continue this discussion for another half hour or so, but we have some other things on our agenda. Really appreciate you coming by. And, one thing you could do that would be very helpful is if you have some links of reports that you have on these matters that we could send out to the board, that would be very helpful. if those are available yet.
Sure. We have manuscripts currently in review. We are cautiously optimistic that they will be in print.
Okay.
And when they are, we can make those available. I don't know if there's any kind of intermediate products that we've produced that would be useful.
If you have any technical reports along the way, for example.
Yeah,
Okay, well, when
yeah.
that happens, we
And-
have an interested audience.
And you guys have my contact information, so if there's any other information that I can provide, I'm happy to talk to you more about it. Thank
Great.
you.
Thank you very, very much.
We don't usually clap, so.
Yeah. That's how good this talk was. I only wish we'd had another half hour or so for discussion. Let's move into thematic reviews. First one being comprehensive monitoring. So, Vince, you want to take over this discussion?
Well, just to kind of bring us up to date, can everybody hear me? Is this working? Yeah. With the last meeting and then through our teleconference, we've developed the prospectus. It was put out for review. We have gotten some comments back. We're hoping to continually get them back, and we'll keep on addressing them. Yesterday we met, and Aaron Forsman and Steve Culberson came and talked with a small subgroup of us that are doing this. We're very pleased that we are getting responses back. We've certainly gotten very lengthy responses from some groups, and very, very useful as well. So we will use these in modifying and updating the prospectus as we go along. We are at the stage of now starting to draft the request for proposal. That will be done through the Delta Science program. And so I think we're following our schedule, which is really only, a few months into it, but I think that we're really making good progress. And again, thank everybody that has commented, and we look forward to more comments.
So, any comments, questions? Oh, come on now. Is this just fine, or just move along? Okay. All right. I thought we'd have something more interesting going on with monitoring. Do we have to have you fill out a blue card? No. No, okay, good. Green card. Green card.
I'm good there. Stephen Culberson, IEP lead scientist, and I stepped forward basically to address a couple of things that Vince had mentioned. And like Dr. Collier, I have some limitations. My wife does not let me touch the checkbook. I'm a scientist, not an accountant. So when I address issues of funding and availability of that for the IEP, I have to sort of check numbers very closely. So I appreciate the comment, 20 of $23 million are spent on compliance monitoring. I concur with many of the observations here, but I would be careful about proceeding along any of those lines without further information, and we'll of course make our programmatic documentation available to you as you do your review. As people have mentioned, some of that compliance monitoring is very carefully specified, and some of it is very loose. The biological monitoring tends to be a little bit looser. One of the things I'd like to remind people of is that particularly the long-term data sets upon which many of our initial impressions about performance of the ecosystem, the estuarine ecosystem, they may or may not be what people think of in terms of compliance monitoring. But remember that today's focus and the focus for the next few years may differ greatly from the focus that we had 10 or 12 or 20 years ago, and may be very different from the focus we have 20 years into the future. And without at least some consistent data stream against which to check what we think we know in specific cases, we may lose sight of the overall trajectory of the estuary as a whole. And there are people who may regard those long-term monitoring data sets as compliance monitoring, and there may who say, that's a lot of wasted money, we don't need to know that anymore, that's not part of our compliance monitoring. So there's a subtlety there that I think we've talked about and then you need to be aware of, and I just wanted to make sure that that was on your minds.
Yeah, thanks, because it's good for the whole committee to hear this. Thank you.
Thank you. All right. Do we need any further action on this? Reiner, you're happy with everything?
Yes, I am. We have a path forward, and we are getting in gear to draft an RFP. I cannot tell at this point at what stage I can share it with the smaller work team. My hope is that by the May in-person board meeting, there can be a discussion about the draft RFP, and in the meantime, probably some back and forth with the smaller group.
Okay, good. Yeah, as the speakers were speaking, I was having visions of statistical optimal control and optimization of monitoring designs to answer questions on population estimates and things like that. So I don't think we'll get to that in the first contract, but maybe that's something we can work towards. All right, well, let's move on then to Delta as place. Dick, you have a report. We have a report.
Yes. So the report we have printed is a month old, and we've been working on this for 33 months, and we hope to move it past working on it more. The last draft that I think, sent public had some 40 pages. We're now down to 9 pages, and finding that more comfortable. Since February 14th, Valentine's Day, we have included material that Jay Lund has provided, so it has more of a levee emphasis and some agricultural emphasis that it didn't have before. John Weens was kind enough to say this really needs at least a diagram, and so it's not just words, and the diagram was circulated and stimulated Steve Brandt to say we need to modify the text to complement the diagram, and that can be easily done. And some other good comments came out of the diagram also. I have a few other comments from the board to soften the language here and there, or to just make it shorter, because I've said, or some wording has crept in that isn't necessary and isn't constructive, and so that will be taken out on several pages. But otherwise, I think we're in a position where, with modest changes, maybe an hour's work, this is ready to go, but it's not the document that's actually before us that's ready to go. I think, Martina, was it posted on the web as the new document, or? Not yet. Okay, so that's what I wanted to confirm.
Tracy?
So my view of this, and having read it, I sort of was out of the loop for the first 30 months anyway. And the last 3 months I've been more involved in reading and providing comments. I think it's ready to be done. If we had a draft last on Valentine's Day and now it's St. Patrick's Day, our next major holiday is probably Memorial Day, and I don't think we should wait that long.
No.
Right.
So I think we should, I would suggest that we move to approve it with final editing from Dick and call it done, if other people feel the same way. Or we can wait till 4th of July.
No, please.
Do we need to have the current draft posted with some period of time?
before we finalize it.
before we finalize it?
Yeah. I don't know. Gosh.
Lauren knows.
Yeah.
Yeah, I was just going to offer the suggestion of what you recommended, but maybe provide it for the April teleconference and actually finalize there. Unless that's, I was going to offer tax day as the holiday, but.
Tax day, we'll do it on tax day.
That would be the marker.
That's a holiday now?
Well, I don't know. But I think, and I'm not sure how significant the changes are, I have to admit.
Yeah, I described them, and they are no more significant than what I described. So there's, the substantive improvements are including the levy and some agricultural information.
You're probably right, though.
I think procedurally that's probably
Otherwise,
right.
it's editorial, pretty much. I mean, John Ween's figure helps, but it's not, nothing was changed in the process. It improved the presentation significantly.
Since levies and agriculture are two of the biggest issues in the Delta, maybe we should wait until, to hold the final vote until the April meeting, which will give us plenty of time to do that hour's worth of work and post it.
And then post it, we can always take action on these things, but post it clearly as an action item for
Yes.
approval and let folks know that
Sounds good.
for the April teleconference.
Vince and Liz?
Dick, I don't want to sound like I'm suggesting more work, but, the material that was taken out of it, I found really interesting that Brian had contributed and you had worked on in terms of geology and, the Earth history. The discussion at one time was to put that in an appendix, but if we don't do that, could you at least consider maybe taking that material and putting it in one of the Stewardship Council publications as a, I just hate to see that stuff lost. It's so interesting, and I'd never seen that anywhere else in terms of put that simply and that straightforward. So that's all I would ask. If you don't want to consider it as an appendix, perhaps you would consider something like that.
It was incoherent combination of material. We tried to bring some coherence between the topics and make some sort of a continuous report, but it really was not coming out like a continuous report. At least some of us are thinking about writing perhaps a San Francisco Estuarian Watershed Science article, but that would really be stressing the human natural systems approach, that it's amazing that in a delta so transformed by people and so those transformations so affecting people that we do not have a coupled human natural systems line of thinking really going on here. And so that one would be fun to expand upon and give examples. Well, Liz has already picked up the examples from other deltas and other regions where this kind of research is being done. So we should at least make that contribution. The geological material, I agree. Brian did an excellent job of very concisely and sort of 3 time periods that are really critical to understand, saying here's what we need to know. And that, I would hope somewhere it could be a standing document.
Yeah, could we perhaps consider, since the thrust of that would be very different than the thrust of this report, perhaps we should just consider writing that up as a separate technical memo or
And then Joy
report.
and Michelle Stevens are pretty sure they're going to be writing an article together on the indigenous knowledge material. So we too do not want it to be lost, but it just didn't seem to hang together in the report.
Well, maybe, Liz.
Sorry. my comments are not about the report per se, but thinking more about the next steps following the release of the report. So yesterday, Jessica Law distributed the 2017 priorities for the Delta Stewardship Council, and one of those priorities is enhance Delta as place. So I think, release of this report at this time, particularly with the Stewardship Council also thinking along these lines, is important. But in recent years, as we've produced these reports, we've also thought about rollout strategies for them and how to sort of make them be more effective upon their publication. So that's just something I'd like the board to think about as we near release of the report, to also think about what actions we might want to take as part of the rollout.
Thank you. There has been a tremendous development and interest in Delta as an evolving place in the period that we've been struggling with this report, and I think it has been somewhat interactive, that we've helped stir the pot at least. And I was especially pleased to hear Director of Department of Fish and Wildlife Chuck Bonham on a 13th Water Law Conference at the University of San Francisco in January make some very strong comments on the need for a sense of the Delta as a place, and that we really are not in a position to make wise decisions until we understand this topic. So it's, we're late.
We're late, but we're ahead.
But I think our process has been part of the process.
Yeah, this might, in terms of rollout, Liz, you bring up an excellent issue. This might be a good topic for us to have an op-ed piece as well, actually. We'd have to actually figure out exactly what we want to say. Of course, that's the most important thing, but I think it would be a good topic for that. And that might be a good way to roll out some of the ideas.
So the path forward.
Dick, did you have any thoughts on rollout?
Well, I think we were talking about an op-ed yesterday about a very general topic in which we didn't actually have to say, pass any judgments. But in some ways, this third leg in the triangle has been not given as much emphasis, or the third leg of the stool, and there is a little bit something political about having an op-ed saying that. But we may be able to find a way of just saying it, strictly as a scientific fact, or, just from the legislation to what's going on and what's the base of knowledge.
Yeah, I think there's enough things to say that would be useful in this area that would offend all sides equally, if that's being apolitical, but yet useful.
Yeah.
Anyway. Well, with that in mind, let's have the report tidied up, and then we'll post it, and then we'll put it on the agenda for a action item as a vote at the April teleconference. Very good. Thank you very much and Vince, let's have some conversations with board members to see what we might do with that other useful long-term perspective material, which I thought was very, very good as well. Water quality.
So we had, I think, a really productive meeting yesterday. Vince, Liz, myself, Tricia, and Val Connor. We started off with a pretty nice detailed outline that Val had helped us pull together. We discussed that at a fair amount of length and modified it, and now what we're going to do is, over the weekend, I'll take an initial cut at assigning writing assignments to the three of us primarily, but some to Tricia and some to Val, but their time is more limited. And we're just going to start drafting portions and seeing how well the pieces fit together in what we're conceiving of as an outline for the water quality review. Because by now we've done a number of interviews, we've done the questionnaire, we've collated the results, we've pulled together, Tricia has really helped a lot in pulling together the bibliography, and Val has done a great job of helping us make sure we have access to probably not all, but a great deal of the gray literature on the subject in the region. And now we're going to sit down and start writing. What would we add to that, Vince or Val? And they made me promise not to assign everything to the two of them, which I thought that was an outrageous abrogation of my authority.
I thought nothing would be assigned if the first initial was a V.
No, it was a T, not a V. Anything else?
All right. Any public comments on any of these subjects? We're just tearing through this thing. When do you think you'll have a draft?
Well, now that wasn't on the agenda. Yeah, let's think about, next leap year, that's a good holiday. I honestly don't know. I think we just have decided it's time to start writing, and once we start writing, we're going to see if it's holding together like we think it will. So we'll update you. We'll update you in April as to whether people have actually put pen to paper yet. But
Fair enough. We have a couple of other things that are on our in that frame as well. Okay, preparation for the meeting, April 13th, 9 to 11 teleconference, and then the May to 11 to 12 in-person meeting. We have comments from WaterFix that'll be coming in. We should have organized by then. Right, Martina? You're going to, this is on the list. We had the 24th was when the comments were going to be due from people for the WaterFix, I think. That's what we decided yesterday. You guys should take notes, right?
I have notes, I know you talk about you are taking the lead on the long-term effects yourself.
And I'll take the lead on long-term effects.
Mm-hmm.
And John and Dick were going to write little bits, and then we're going to go to you, and then that was going to reflect out to the board members for all these parts.
Right, I think the discussion was everybody will kind of pitch in for the kind of high level, or I think the missing opportunity kind of thinking, and then,
But
John and Dick will kind of...
And Dick, they went to some trouble to volunteer, and I intend to take them up on
that. And I have Tracy as well. Are you part of that?
I think I was.
Yes, I think so. So I have, you as well. Okay.
That's on WaterFix, and so hopefully we'll have those comments in by early April, and maybe we can even assemble, shake it down a little bit, 3 or 4 of us, before that meeting.
Lead scientist.
Lead scientist, yes. We should probably have a closed session with a lead scientist recommendation vote. So I'm thinking we'll probably need, let's think about having at least 20 minutes for that. Maybe we can update that closed session estimate.
So now the teleconference is just 2 hours on the calendar.
Yes, I understand.
Is that
but this is one of the most important things we do.
doable? Okay.
We'll have, hopefully we'll have a draft of a white paper recommendation, review memo.
Eco-Restore?
For Eco-Restore, yeah.
Science Action Agenda, so you will receive the material prior to the teleconference.
On that one, I thought we agreed individual comments would come in to Joy and myself by the 31st, right?
Right.
And then we'll see what we can pull together by the teleconference.
Right. And hopefully we'll have something we can just vote on with a little bit of luck. Then we'll have the final version of Delta's Place to vote on. We'll have some detailed.
It's going to be all action all the time.
You bet. And then we'll have some discussion about what we're going to do at the May meeting. Looking out for the May meeting, we're going to have the water fix presentation. Oops. Looking at the water, we'll have the water fix presentation in the May meeting. And some more detailed discussion based around whatever draft we have at that point. We might even on the second day be able to vote on a draft of the water fix review, in my optimistic delusion. But let's try to push it forward anyway.
Jay, on your April meeting, the Science Action Agenda draft will probably be coming to you for consideration. Okay. So that
how much time would you like to have for presenting that to us?
I think probably 10 or 15 minutes would work.
Okay. We can probably squeeze that in. And we'll have to talk about how, you want us to review that, right? So we'll have to talk about what timeframes you would like that to be done, and then how we're going to organize ourselves to do that. And by the May meeting, we might also have some conveyance, storage, and operations topics to be presented to us, plan amendment for the Delta plan, as Tom Zuckerman so usefully foreshadowed for us yesterday. What am I missing?
The State of a Delta Science retrospective.
In May?
Yes. In May.
Good, very good. What else am I missing? Dick, is your cards up? Okay.
This did just spoke.
Okay. And we'll see what else has been put on our table by then. We should also be starting to think about field trips and Delta meetings in a place, meetings in a place in the Delta. There we go.
I think we decided right.
But we need to fix a date for that, and then build some field trips around it, I think. Right?
And we were also talking possibly in July to have another retreat like the one we had 2 years ago, and sort of just to talk about not only our process but more about what are some of the major reports we need to start putting on our agenda.
That's a good idea.
And also, we talked about, we didn't talk about it too much, but whether or not we want to also solicit external feedback as to how we've been doing over the last 2 years, because it was 2 years ago that we asked a number of stakeholders, are we any good to you or not? And we actually got pretty positive feedback and some good suggestions, and it may be time to do that again, or maybe it's too soon. But
That's a very good idea. They're all going to come back, boy, I'm glad that Steve is the new chair, and that Liz is the chair-elect.
yeah, but none of them are going to be as glad about that as you are.
Right. Any other thoughts, comments? Any comments from the public on anything at all? Anything you want to say? Sam's not here. I was going to make him give a speech. All right. Well, that's all, folks. Oh, wait a minute.
I just want to ask a clarifying question on the due date for the monitoring perspective. I believe the new email stated it was March 31st. And as I heard, the RFP will be drafted somewhere in May. Is there enough time for the public comment and review and then the RFP draft? So I just want to...
I don't know, does the RFP get reviewed?
By you, yes.
Okay.
No.
Not public.
By us as individuals, not necessarily us as a vote.
Thank you, that's all I meant.
Great. Any further comments, questions? Okay. Wait a minute, Tracy?
So the one thing we talked about yesterday in the monitoring working group was, yeah, we will take comments on the prospectus until March 31st, and then we'll revise it, and that'll finalize it. But so the prospectus itself, the review comment period is until March 31st. Is that right, Vince? We decided to wait till then before we do any revisions to it, so. Yeah, okay.
Okay. Thank you very much. We'll end early. Happy St. Patrick's Day.